I'm going to talk about the design of complex adaptive trials. We've heard about adaptive trials. We've heard about very, very complex adaptive trials. We've talked about the need to do it differently, the Titanic analogy and where we're going. And so how is this done? How do we design an adaptive trial? How do we design a complex adaptive trial? Okay. First, let me talk about the promise of adaptive trials. Why is it that the excitement of adaptive trials? The idea is that you learn something during the course of the trial that had you known that before the trial started, your design would have been different. Had I known that high dose was unsafe, I wouldn't have put it in the trial. Had I known the low dose has very poor efficacy, I wouldn't have had it in the trial. Had I known when I put those two drugs together that it has a really strong benefit, I would have put more patients on that arm. The idea, the promise of an adaptive trial is that we learn that during the course of a very long trial. We adapt the trial so it takes those decisions. It drops unsafe arms. It puts more patients onto beneficial arms. That's the hope here, is that we can have the trial morph into it and learn that during the process of the trial. That's the promise. Now, what do we mean by a complex adaptive design? And this is my definition. I think Pecora used a similar definition in their adaptive trials guidance, but I'm going to refer to it as one in which the design is complex enough that you can't analytically calculate its performance characteristics. You can't analytically calculate power, type 1 error, the probability this arm wins, the average sample size of this. And this works nicely because I'm going to bring up the notion of simulation. It's going to need simulation to characterize the design well. And so if you're doing a group sequential method, we understand power and type 1 error. You can write in the protocol we're using an O'Brien-Fleming boundary at 50%, and this is well understood. I don't think it's as well understood as it should be, and simulation is still valuable. But once you get to response adaptive randomization, enrichment designs, seamless shifting, arm dropping, combining arms together, putting multiple drugs in the same trial, and doing all of those in one trial, you now need other mechanisms for understanding, is this a good design? And so this is my field of complex adaptive trials. So what does it take to build a complex adaptive trial? The first thing is that Dr. Getz talked about, Ken Duck talked about, the customization. Adaptive trials, in my view, is customization. That we don't say, oh, I'm going to use Dragolin's design number 7. He has far more than that. But this isn't the idea that we have hammers standing around and, oh, we're going to hammer that nail with this design. That the notion of an adaptive trial is customization. There's a unique solution for every trial, for every patient population, every drug. When you go in, we don't recycle designs and say, oh, let's bring this design in. It might not be right for that. And answering the question is that design right for that, is part of the process of building complex adaptive trials. So we've built many complex adaptive trials and never the same one twice. They have things in common, but it's not like we're recycling designs. The notion here is customization. So how do you do this? What's the process of building these types of designs? So the initial steps in the interaction, and actually my bullets aren't in a very good order here, so I'll bounce around a bit, is understanding what is the drug, what is the device, what is the medical, what is the disease, what's the landscape. This is always a critical part, and this may be an interaction or two to really delve in and understand what they are. And then going back to the first bullet, why are you doing the study? You can't build an efficient design if you don't know what the finish line is. I think Yogi Berra said something very similar to that. If you don't know where you're going, don't be surprised when you don't end up there. So are we doing it because phase 2 comes after phase 1? Which I've been told before. Are we doing it to understand the most effective dose? Are we doing it to make the go, no-go decision of phase 3 as efficiently as we can? What is the role of the trial within drug development? What happens after this? Do we do a second phase 2 trial? Can we incorporate under what outcomes of the trial we're building will need a second phase 2 trial? Under what outcomes we would go to phase 3? Can we incorporate that? What is a failed trial? It's hard to build a trial efficiently if we don't know what failure is. What is a successful trial in building that? Can we incorporate those? And as you're going to see, we're going to simulate the trial. And can we take every simulated trial and attach, is this a failed trial? Is this a successful trial? What would happen next becomes a critical part. These are hard questions to answer, but a critical part to building a good design. And we're working under the framework that the traditional model doesn't typically ask these questions. We run the trial and then we ask these questions because they're hard. And the question is, do we have the right answers? Do we have the right data? Do we have the right trial? Well, we find that out later. The critical part of this is then what's uncertain about our drug or device. If we know everything about the drug, we know the right dose, we know the right frequency, we know the effect size, we typically can design the right trial. But most of the time we don't know these. What frequency should it be given in? What's the duration? What are the device settings if it's a device? What dose to use? What is the right population? Do we know what the right population is in that circumstance? Do we know the effect size? Many of this stuff is uncertain. So trying to capture and understand what are the uncertainties that our design can learn about that might help the adaptations. Remember, the promise here is that we're going to learn something that's going to help the design. So what uncertainties can we learn about during the process that might allow our design to evolve more efficiently? And of course, one of the questions we commonly ask is, suppose I tell you the design fails. What do you wish you would have done differently? And if they say, oh, I know we got the wrong patient population, I would have included this group, then maybe we can do that in the trial. If it starts going negatively, we add that population, or we change it in that way so that our single silo trial isn't a failure that results in very long time or an unnecessary trial being done after that. Okay. So as a statistician, a key part of this is trying to understand the uncertainties. What is it, a dose-response relationship? Is it duration, frequency? All of the uncertainties to the medical therapy. And start to think about, if we start to learn about these, what adaptations could we use in the trial? Could we allocate differently, which includes dropping arms, adding arms in the setting, changing the randomization probably to favor some arms over others? Do we refine entry criteria? Do we drop tumor types? Do we add tumor types? Do we restrict in an emergency setting that you need to be treated within 3 hours as opposed to 8 hours? These are things that could happen during the course of the trial. Maybe we extend the trial. We don't know the answers yet. Maybe we shorten the trial. We know the answers. And these are the adaptable pieces. These are the scenarios we run through, and then we create a skeleton. We haven't simulated anything. We say, okay, how would the following design look like? And we say, okay, we'll do response adaptive randomization over the different doses, and we run that, and this is how is this a good design? And we share it with the team. Does it seem like it answers the questions? Well, no, we need to have a population aspect to it, that we might have different results within these two patient populations. And then we go back and we think about the design as well, and we come up with a skeleton of the design. We still don't know in a complex design things about type 1 error. Many times in a complex design, type 1 error isn't even a clear question of what it means. Because there are many moving parts, power is a complicated issue, and then we say, okay, we now need to simulate the design. We need to create a flight simulator of the design, simulate it, look at the results, and say, is this a better state than we're in now? Would we go to phase 3? Would we approve this? Would the clinical community accept this? Whatever the questions that we originally had, does this answer it? And then we go through this process of iterating to create a better design. So simulation is something that statisticians are quite familiar with it and love and use it a great deal. But we've had a lot of difficulty explaining the role of simulations to teams that haven't used it before. So I'm going to go through an example trial. I consider it a fairly complex trial where I'll explain the role simulations play in this. This is an ongoing trial. It's part of the Adapt It project, which is funded by the FDA and the NIH. It's really a cool project, unique in the sense that we interact with the FDA, clinical teams together in building these trials. We work on 5 trials that are in the Neurological Emergency Treatment Trials Network. So they're therapies that are used in the emergency room. It's cardiac arrest, stroke, spinal cord injury, epileptic seizures, I believe. These are the types of things that are treated in the emergency room. And so we're bringing adaptive technology to these. And part of the grant, which is really neat, is that there are individuals that are studying the process of bringing adaptive techniques to this application. So what works? What are the barriers? And there are surveys done, and we're actually being studied in the process. So we have approval to study us. So the ICECAP trial this is under construction. This is not final yet, and so this is part of see how the sausage is made. The ICECAP trial is bringing hypothermia after post-cardiac arrest, coma. So this is an individual suffers cardiac arrest, in the field is revived, the heart is brought back, the individual survives the cardiac arrest, is brought to the emergency room. The idea is that then, and I'm not a doctor, but I'll play one here, is that a number of cascading negative things happen that are brought on by heat. And heat is an accelerator of this, and the notion is that this is a very negative thing. So the idea is that you treat them, you put them under a hypothermic state, and it could slow down or prevent these negative cascading things that can happen during the course of the trial. And this can be beneficial to the long-term neurological status of the patients. There have been small trials done before, a number of trials with quite promising results for hypothermia for the treatment of post-cardiac arrest, but it's not FDA approved. The medical community believes that this works. So it's the standard of care in many hospitals, but it's not FDA approved. So there are many questions and uncertainties, and so part of this is how long do you cool them? What is the optimal duration? And it's a scale of maybe 12 hours to 5 days that you cool them. What is the appropriate time? Does it work? What's the relative efficacy? And then later, through the process of simulation, and I'll talk about what came out of this, is that different cardiac arrests may bring about different needs for duration of cooling. So there may be a population effect to this. The fixed design when we first came in was where they would go in with, and I'll use a placeholder at this point, 300 patients on 12 hours, 24 hours, and 48 hours of cooling. That's sort of the fixed design. So the first thing we want to do is understand that and what might be the shortcomings of that fixed trial. We're going to bring adaptive technology and think about doing the trial differently. Think about that and what are the benefits and negatives and benefits of that. So the first thing we might do is simulate the fixed trial. And let's ignore for a minute the y-axis. I'll talk a little bit about that. But think about high values here as being good. Here might be the results of this fixed trial, where we put 300 patients, that's the bars along the bottom. The red circles and red squares there are the point estimates and the confidence intervals that come out of the trial results. So this is a single simulated trial and the result of it. And this is something that actually might be a very likely outcome of this trial. So then we say, is this a good trial result? Does it answer all your questions? Do you wish you'd have done something different if this is the trial result? Well, this was looked at, and a lot of people say, well, does the curve go up and does it continue up into 36, 42 hours and then come down? Does it go up after 12 hours and maybe 18 hours is optimal and then it's down after that? Is it flat? Is the duration response curve flat, and does this answer the question? It's not clear from this trial. So if this were the resulting trial, does it get FDA approval? Does it change clinical practice? Or are we stuck in a similar state of nature after this trial? So we then say, okay, we would have loved to, in this trial, have seen what happens on 36 hours, what happens on 30 and 18 hours. We'd have loved to have seen what would have happened in there during the course of the trial. It's looking like 24 is good. Maybe we could have taken patients off of 48, put them there, or off of 12 and put more patients there to better answer the questions. And so that's what we're going to try to do with an adaptive trial. So the first thing we're going to do is we're going to start the trial the way the fixed trial was set up, where we'll put 50 patients on 12, 24, and 48 hours, and then we're going to analyze the data. The idea, the promise of the adaptive trial is we're going to learn something during the course of the trial. So we're going to do an analysis that says, what do we know about 12, 24, and 48 hours? And then we're going to say, should we change the allocation? So we're going to allow randomization to the durations from 6 to 96 hours. That's the uncertainty here, and we might need to go bigger, we might need to go smaller. So we're going to allow allocation to vary after the first 150 patients. Then we're going to continue enrolling patients according to our new scheme, analyze the data 50 patients later and say, what have we learned now? How should the trial change? And we'll continue that. We'll continue to ask the question, should we stop the trial? Have we learned enough that the trial should be over at this point? And so I'll start this process and show you a simulated trial where our endpoint defines the modified Rankin score at 90 days of 0, 1, or 2 as a success, 3 through 6 is a failure. That's where we started. So I should have said this, thank you, that everybody has to be cooled. The standard of care at these centers is that everybody is cooled. So everybody will at least be cooled for 6 hours. This avoids issues around waiver of informed consent. Everybody is randomized. Everybody is cooled initially. So we will not prevent people from getting that. And that was a part of these studies being a part of it, is they believe this works. So learning about the efficacy of cooling is going to come from the duration response model. So here's Roger's slide of what most adaptive trials look like. And now we can put this skeleton, all it is at this point is a skeleton of a design, into a flow chart. We allocate 50 patients on each of the arms. We analyze the data. We ask if we should stop the trial. In my current simulation, it only stops when we get to 1,200. That's a feature we'll add in to decide, do we know enough yet? Then we revise the allocation rules. We change the randomization probabilities. We accrue 50 more, and this continues in this circular pattern. So what is a clinical trial simulation? What we do is we take that skeleton and we put it into a box. This is now a well-defined design that we can put, refer to it as a flight simulator here, that we can take this trial for a flight. In order to do that, we need assumptions about how do patients behave. So we have virtual subjects that we put into the trial. And we get to play Mother Nature and change that in different ways. It doesn't work at all. It's negative. It has a strong positive benefit that wanes at the end. We get to take it on lots of test drives. We also have execution variables that are critical to the success of the trial. The accrual rate, the dropout rate are critical parts that can be crossed with the way we simulate patients. Now, we simulate this thousands of times, and we can look at what happens at the end of the trial. How often do we get convincing results? What is a convincing result? But one thing that came out of this simulation, and came out really the Adapted project, but of doing this for a while, is the critical role that single trial simulation plays. The viewing of a single trial and showing the team, showing regulators, showing DSMBs the process of how the trial goes. So I can demonstrate that here with, this is the first skeleton in some of the first simulations we did. So we create the virtual subjects here, is the duration from 6 to 96 hours. We play Mother Nature and we create this. So patients will be simulating according to this scheme where they range from 50% success rate to a 70% success rate, and there's a plateau after 48 hours. We can then look at, over 10,000 trials, the average number of subjects that go on each of the durations for the different maximum sample size. And 1,200 is the green one in the middle, this sort of middle of what we're looking at, but we're trying to make this decision as well. So that's the average of 10,000, and it looks really good. We're putting most of the patients right in the optimal area. The probability of determining that that's the best duration is going up with dose, as we suspect, and it looks really good. And we make sure that this one isn't picked with larger sample sizes. And this can be deceptive. The point to these is they look really good, and then we share with them single trials, and they said, wait a minute, I don't like this. And so this is what happened. Here's an example of the first interim analysis of the trial. After 150 patients are enrolled, we have 50 patients on 12, 50 on 24, and 50 on 48. And we make this process look like they're a monitoring committee that are looking at this simulated trial. Exactly what will be seen during the course of the trial. We show in open red circles here what the proportion of successes were in the trial, and here at 48 hours it's covered up by the solid one. And that's showing the data estimated value, and then the solid red circles is our statistical model estimate of the duration response model with the error bars around it. We're now looking at, okay, based on this, what's the probability each duration is optimal? And we create new randomization probabilities, and those are shown in the block on the right for the next 50 patients. And the trial proceeds to randomize those patients. The second look, we've added a number of patients right around 24. It's looking good. And the trial continues every 50 patients, adding subjects. And then we got to this point and said, okay, this is what the data look like at this point. And you can see the average number of successes here is looking really good in the range of 24 to 42. And then it looks like the dose response comes down, and then there's a certain amount of uncertainty in the right-hand tail. We're using a Bayesian smoothing spline for the dose response model. The clinicians and the team didn't like this at all. They looked at this and they said, if we were on the data safety monitoring board or we were looking at this, we wouldn't like this at all. We don't believe it's possible the duration response goes up, and as you cool more, it comes down and then it shoots back up. This is biologically implausible in how it works, and we wouldn't put patients up here. We would continue to put them in this region, and once it starts to come down, this is not acceptable. We know more than allowing this to come up. So it's a really important part in the process that now this customized solution can't just throw a flexible model in here. It's got to recognize what we know about the idea between duration of cooling and response. And so we have to build a different statistical model to get the right solutions to that. It resulted in an unrealistic dose response curve that escalated way too quickly, And it also de-escalated too quickly. It was too flexible within that setting. Here, they wouldn't want to open 6 hours. It looks like there's an increasing duration response. We need to go into the middle. So now, what became a critical part of this is in this flow chart, there's this part of the statistical model and the allocation. And this is now where we need to go in and change the model. So we go in and we created that the curve can only have this behavior. It has a region of going up, a region where it plateaus, and a region where it comes down. It parametrically has to fit within that, and we created a model to do that. We don't know where our durations are, and so we allow it that our durations of 96 hours may all be in here and it's increasing the whole way. It may all be in here and it's flat. It may all be in here, or it may span the range, or it may be in here. That's the uncertainty of where we are, and then the general shape, but it has to follow this pattern. So we added in that model. We also added in a model that uses 30-day MRS to predict modified Rankine score at 30 days to understand the relationship between 30 days and 90 days to speed up the adaptive design's learning. So we built models to do that during the process, and we don't know what that relationship is, so we built a dynamic model that can learn about that relationship during the course of the trial. Then we share with them, okay, here's our updated simulations. We also, by the way, through the simulations, they changed the endpoint. They didn't like 0, 1, 2 as success. We use a weighted score over the modified Rankine. That they didn't like 1 for 0, 1, and 2, and 0 for 3 and bigger, so we use a weighting system for that. We also dropped, they discussed a great deal about how the trial went. We dropped the 84 and 96-hour duration, that wasn't going to work. Part of it was the emergency room situation, holding people in that for that long, and biologically, they didn't want to go that far. So we dropped those from the simulation. So here's a new simulation, and I'll add in here that the probability each one of these is the optimal arm. So after the first 150 patients, this is what the curve looks like. And now you can see it has to be this inverted U, and we've got data that are consistent with that. And so now this is the probability each duration is optimal, and we use that now to simulate the next group of 50 patients. So I'll go through this fairly quickly. The movie of a single simulated trial. And now at each one of these is okay, if we brought this to you as the data safety monitoring board, as a team, are you okay with what the design is doing? Did we do something wrong? And they looked at it, okay, this is where we would allocate now, this seems reasonable. Sometimes they benefit a great deal from understanding what the model thinks. And it passes through, and notice it never opened up 60 or 72 hours because it always looked like it had reached the plateau, and we know it can't come back up. So it passes through, and we show them this is the final result of the trial, looks like this. The result of the trial is that 30 hours is the winning duration, is the highest probability of being optimal. That would be the winning one. And it also is better than smaller durations, which would be the primary endpoint for an indication that this works. It has an increasing duration response. And in that, and by the way, the black in there is Mother Nature. It's what the assumption we made to simulate this trial. So this particular simulated trial got the quote-unquote right answer. It found the right duration. It put patients within that region, and it did well in that circumstance. And then we take this through multiple scenarios. Here's the exact same assumptions we just used. The average number of patients allocated to each of the durations during the course of it. And notice we almost never open up bigger doses. The probability of each one being the winning duration in the trial. And then we take it for different kind of test drives. Suppose the effect happens earlier. That now in this circumstance, 18 hours is optimal, and then it plateaus. Well, it puts the patients within that region, and it goes a little bit higher to see if it's better, but then it goes back down, and we do a really good job at finding the right duration. If it happens much later, the design reacts really quite well to it. We almost never open up 6 hours, which is what they didn't want to do. Puts the patients in the right region. And now, suppose you didn't see those, you would want to open up 60 and 72 because of the increasing effect, and it does that. And it does that quite well in the different trials. We get the right answer quite well. And now we've taken it for a number of different test drives. We've done many more than this. We look at what happens under the null hypothesis, how well does it behave. But a critical part of this was the single trials we went through. So the role of simulations in building the trial, it's an incredible learning tool for the team. I can go through and give them the flow chart. I can tell them what it does. They don't understand it until they've been on a test drive and they see what it looks like. You can share it with regulators, with the funders of the trial, the data safety monitoring board, so they understand what the trial is supposed to look like, what their role in the trial is and what their role isn't in the trial. Operations, you can hand them the simulation and say, this is the trial, and it's ready to run. They know exactly what's supposed to be in there, what data are in there, what the models are. You actually have the programs already ready to go. The process of this allowed us to find the right models, the right measures of success, asking what would be convincing to the medical community. What happened, what was really neat is the last meeting we had for this is they got to the end and they said, okay, if these were the results, my first question is, what does it do in the different rhythm types? There are two rhythm types, shockable rhythm and non-shockable rhythms. Is it different in those? It was the first question they had. So we said, okay, can we answer both of those questions? So the new update is going to be to have two populations with different duration response curves in the same trial to answer those, rather than doing siloed development, which is a critical part of seeing what the trial looks like, seeing what results look like. Yes? Okay. So I will end there. I think I said all of this in there. Okay, thank you.